I'm Susan Rosenberg. I'm a molecular biologist at Baylor College of Medicine in Houston in the United States. And I'd like us to think about and discuss the molecular biology of the ability of cancers to evolve. I think this is an interesting topic because it can give us some ideas for potential preventions and therapies for cancers. The viewpoints, the hypotheses, and ideas that you'll hear about today, these are mine. They are not meant to be a recommendation or a statement from Baylor College of Medicine, where I work. So I want to, before I talk to you about problems and science that I think we could address, scientists always live on the border of what we know and what we don't know, looking to find out the bits that we don't know yet. And because of that, it can sound as if progress has not been made. I want to acknowledge that there has been some pretty wonderful progress in uh, in understanding and dealing with human cancers, particularly women's cancers. So, for example, cervical cancer can now be prevented by a vaccine. And even for folks who are unvaccinated, if they have regular checkups, early detection and surgical removal can cure it. I think that's a bit of happy news. So, cancers actually come from a process of evolution. And I'd like us to think for a minute about how that works. The idea about evolution is that you start with populations of, could be cells, could be individuals, that change. They can get mutations in their genes that can give variable populations of cells. And inevitably, all of those differences will give some of them the ability to proliferate faster than others. And the idea is that through many, many repeats of this process over time, tremendous complexity can evolve, like new organisms, but sadly also cancers and bacterial pathogens who are better at defeating our drugs. I'd like us to focus on this part. How do these mutations happen? How do these cells get the ability to evolve? Why this could be a useful place to focus is because if we could really understand the molecules and the processes that let evolvability happen, we might be able to affect early detection and prevention and potentially new drugs that could let us deal with cancers. So, for example, if we understood very well what cells looked like when they were going to make mutations or before, we might be able to use those signals, those cues, as a way to identify people whose cells are a little too evolvable that we might want to keep an eye on. And then thinking about drugs, all of our current cancer therapies, but this is also true for antibiotics that we use against bacteria, they are all drugs that kill cells or stop them from proliferating. And the problem with this kind of drug, they're great drugs, but the problem is that there will always be some number of cells who don't get killed. There will always be some survivors. Among those survivors, mutations can occur and they can even be stimulated to occur. So at the same time that we're giving useful anti-cancer drugs, we could be stimulating production of mutations that will drive the resistance to those drugs. I'd like us to think about a potential different kind of drug that would actually target and try to slow down the processes that generate the differences between cells that allow evolution to occur. For example, drugs that would slow down the formation of mutations in cells. If we had those, we could give them with our standard chemotherapies and have less instance of the development of resistance. These don't exist. This is a hope. This is an imagination of what might be possible to do. So it means that one has to think about how do mutations actually occur. And for a long time in biology, there was really not much information, and scientists always will prefer the simplest model. The simplest model that I think we all considered pretty reasonable for a very long time was that mutations are a constant, gradual process, like sand falling through an hourglass or a ticking clock, that just accumulate randomly and gradually over time. I want to contrast that with this view on the right, because what my lab discovered many years ago and into the present is that, in fact, when cells are put into an environment that they're not adapted to, where they feel stress, they turn on gene programs called stress responses, and these gene programs activate genes that protect the cell, but also other genes that turn up the ability to make mutations, and this increases the ability to evolve. When we think about cancer cells, some of these stresses could be too little blood, oxygen deficit, shortage of nutrients, crowding of cells, and even the cancer drugs that we give. So cells increase their ability to evolve during stress. This is a cartoon that Beatrice, the biologist, made several years ago. And what it is two cells discussing with each other. One of them doesn't feel well, and the other says, might be time to flip your switch. And the switch is a switch that turns on mutations, emergency use only, turn on to speed up evolution. So I'd like us to think about two topics today in cancer evolution. One is, could we eventually have drugs that would slow down cancer evolution and potentially prevent therapy resistance? And I will show you a test case that my lab made in bacteria. And then I'd like us to consider DNA damage. Damage to DNA is a very early event that can happen before mutations occur, and could we use it as a way to tell who is likely to have too many mutations? What you'll see is that both of these topics my lab approached in bacteria because we work on the bacterium E. coli, and it turns out to be a really very good model for how cancers work, because the problem of mutations is about the biology of DNA, and virtually the vast majority of organisms on the earth have DNA making up our genes, and it works the same way in bacteria as it does in humans and in everything in between. So I was telling you a little bit about stress inducing mutations, and this is a sort of a cartoon diagram of a mechanism by which this occurs that my lab discovered some time ago and has been working on for decades, and we're still learning more about it. The idea is that when the DNA suffers breaks, if the cell is growing in a non-stressful environment, the repair of those DNA breaks is fairly accurate. Mutations are not made. But if the stress responses turn on, there will be a switch that will make the repair of those DNA breaks mutagenic, and mutations will occur. For the ones we were studying in E. coli, 4 different stress responses were used, the most important of them being the general stress response. So this picture is to show you how there have been many labs working on this in various organisms, but also in the context of human cancers. And what you see on the left are a list of human stress responses that when they turn on, more mutations happen. What you see on the right is when we discovered some of these things in E. coli. And I just want to go through the elements that seem to be in common between multiple different ways of turning up mutagenesis. So first of all, stress responses being the switch, that's all of these on the right, and that was certainly the case that we found in bacteria. Then another point in common is the generation of mutations by making DNA break repair less accurate and more mutagenic. All of these stress responses do that. Then finally, the cells will turn down the proteins that correct errors that are made during DNA synthesis, and that means that mutations happen. So all of these things were happening in E. coli, and they are happening in human cancer cells. It's useful to understand them in detail because these could potentially provide us targets for drugs that would allow us to slow down these processes, to prevent resistance in cancers, and potentially to identify the cells who are very mutable before a problem happens. So it could be a marker of trouble in cells. This is a test case that we made in bacteria for the idea of finding drugs that would turn down mutation rate to slow down evolution. We did this in E. coli bacteria that we had engineered so that when they turn on the most important stress response, they fluoresce bright red. And then we took these cells and put them into growth vessels with a library of different drugs. This was a small library, only just over 1,000 molecules. And the idea is that when the bacteria with the antibiotic go in there, they're going to be stressed and fluoresce. I didn't mention this, but the stressor is this antibiotic, which actually works similarly on bacteria to one of the anti-cancer drugs that we use on people. So drugs that don't do anything, the cells will fluoresce red, but there's the occasional drug that turns down the activation of that stress response, and then there isn't so much red fluorescence. My student used this screen to identify from this small 1,000-molecule library 2 drugs that prevent the stress response from turning on. And then he used them to see if they could reduce the number of mutations that happen in bacteria that are infecting a mouse. How this worked is that bacteria are injected into the mouse, and then those mice are given the antibiotic, and they're given it with or without the mutation-slowing drug candidate. They get this every day for 2 days, and after 2 days, bacteria are recovered from the mice, and we see how many of them have mutations. So the number of total bacteria can be determined, and the number of bacteria who carry mutation in a gene that makes them able to be resistant to even a different antibiotic, an antibiotic this bacterium has never seen. And what he found was that when we looked at the control, and here is mutants per cell times a very large reduction factor. So these are 2 out of 1 billion cells will have a mutation. In the control, if he uses a bacterium that can't turn on one of the stress responses, the frequency of mutants is down. If he uses cells that are unable to turn on the second stress response because of a mutation they carry, mutations are also down. And the 2 drugs that were identified, they turn down the formation of mutations as well as knocking out these stress response genes. So this is encouragement that a similar strategy might be able to work in human cells with cancers. So it hasn't been done. We don't know if this can work, but it's hope that makes us want to try. The next topic I want to tell you about is something that happens to cells maybe even before stress responses, and that is damage to DNA, which can provoke mutations and evolutions. So when DNA is damaged, it has to be repaired by DNA repair mechanisms. Often that process is accurate and mutations don't occur, but sometimes it's inaccurate and mutations occur and evolution, including cancer evolution, can be promoted. Of course, they need to use DNA repair only when they have DNA damage, which makes one wonder, where is all that DNA damage coming from usually? It's easy to think about all the ways that DNA can get damaged by processes that are outside our cells and outside our bodies. Mutagenic substances and conditions that come from the world we live in, like pollution or chemicals or radiation. For most of us, unless we smoke, that's not the case. For smokers, chemical damage is the biggest source. In fact, for those of us most of the time who are not exposing ourselves to hazardous chemicals, our own proteins are causing little perturbations in cells that can provoke DNA damage. So just the bits of cells that are doing all of normal biology, if there's a little too much of one of those proteins, which happens sometimes, they can make the cell a bit out of whack and DNA damage can result. So I will tell you about our discovery of some of the proteins that do this to cells. Why we wanted to look at DNA damage as potentially a flag to tell us whose cells might be too evolvable is because the idea that it would be prevalent. So for example, over here on the right, the specific mutations that cause an actual disease, including cancer, those are extraordinarily rare. Cells who get mutations at all in any gene, even in genes that don't cause disease, those all by themselves are extremely rare. But if most cells have to suffer DNA damage before they turn on the mechanisms that make mutation, maybe that could be something that we might be able to see in large numbers of cells. So it might be a bigger target to be able to identify and find in people. If this is true, DNA damage, it might be just an early signal of whose cells are too evolvable. And this is not something that works now, but I imagine I can dream that we might one day have a test sort of like blood pressure. When you go to your physician and your blood pressure is too high, your risk for heart attack is up, even though you don't have a heart attack at that moment. If we had a test that was that easy, that could be done that regularly, then we could identify people whose cells are having too much DNA damage, presumably they are too evolvable, and then these people we would want to monitor very closely to see if we could prevent their future cancers. In order to do this, of course, we would need ways to do that, to prevent those future cancers. I don't think these are unimaginable. I think they are imaginable. So the example of looking for them in bacteria and humans was the work of another student in the lab. He used bacteria that carry in them a gene that encodes a red fluorescent protein, mCherry, and that gene is controlled by a bit of DNA that will make the gene turn on when the cell feels DNA damage. So what he did was look at every one of the 4,000 genes that E. coli has to ask, when each of these genes is a little too active, a little too much protein is made, do any of them provoke DNA damage? And from the 4,000 genes, he found about 200 E. coli proteins when there's a little too much DNA damage results, and those cells will fluoresce red. Those 200 E. coli proteins gave varying levels of DNA damage, and it was in fact the case that the more DNA damage, the more the frequency of mutations increased in those cells. These 200 bacterial proteins have 284 homologs, or proteins that are similar in humans. So humans have proteins that are very similar to these E. coli proteins, and we thought we should look at these proteins in humans to see if they have an association with cancer and with mutations. What I'll show you, first, this is a list of some of the kinds of proteins that are in there. Very few of them are actually proteins that work on DNA. There are all kinds of proteins in here that do all kinds of jobs. Proteins that are part of metabolism, proteins that live in the membrane of the cell, transporting molecules into or out of the cell. Proteins that we didn't think would necessarily have anything to do with DNA, but there they were. When we look at the RNAs of those 284 human proteins in the cancer data from a variety of cancers. So now these are data from many cohorts of humans who had cancer of one kind or another. So for example, you can't see their names here, but breast cancer, lung cancer, colon cancer, all of those are represented and several others. When those 284 genes are a little too active, we see more mutations in those cancers. So the blue indicates how many mutations are found in the cancers, where bluer and a bigger dot means more. And if there was no association, it would be white, which you see if you look at activity of all human genes. But these 284 proteins that are like the bacterial damage-promoting proteins are associated with accumulation of mutations in many different kinds of human cancers. That's encouragement that this property might hold for human beings and cancers. And for a few kinds of cancers, when those 284 RNAs encoding those proteins are high, survival is worse. So the point of this second part of our consideration of evolution in cancers is that there happens to be a large collection of proteins that, when disturbed a little bit, will provoke DNA damage that requires DNA repair that increases the probability of mutations that can lead to cancer. And they are a different kind of cancer-promoting protein from ones we had thought about previously. But sadly, there are so many of them and so many kinds that a thought that we have is it might be easier, rather than looking for increase of any one of these proteins and the many more that we didn't discover, to look at what they do instead, the crude gross level marker of DNA damage itself. Maybe if we were able to assay that in people, we could be able to assay people to find out who is likely to get cancer, who may have cancer at an early stage when it's operable, and whose cancers might have groups of cells that are evolving resistance. We hope this might be a marker. This is not something that exists. It's a direction that my lab would like to go in. And maybe it could be as simple of a test as something like blood pressure, where when you find people who have a problem with it, then you don't even need to know exactly why they have that problem. You do know that they need to be helped. So I will end here with this picture of thinking about cancer evolvability and why it might be informative and important to understand how the differences in cells are generated that let evolution occur, so that we could detect people before they have cancers, I hope, and how it might be possible to design and make new kinds of drugs that would reduce mutation rate, slow down cancer, slow down evolution, and potentially let our current drugs work without causing resistance.